Okay, so we're going to start with the t-test, that it is analysis for two variables, or it can be even the same variable, but after something happened, because that is a great use for a t-test. That is a great example of a t-test. Imagine that you have the average, sorry, you have a sample of people before going into a diet, and then you have the same group of people, but after the diet. So you want to check with the t-test if both groups are statistically different, if there is change from the first one to the next one, and also if they are related. Do you understand? Like if they are not exactly the same thing, but also if that difference is, if there is relationship between the both, like it's kind of a middle point between, it's not the same, but it's not totally something different. Okay, I hope that you understand. But the definition is that it is used to compare the means of two groups. It is used to compare the exactly like that, if it is statistically different. And as I said, it is often used in hypothesis testing to determine whether a process or treatment actually has an effect on a population of interest, or whether two groups are different from one another. Okay, so that's the definition when you want to test if the groups are different, or if the process or treatment had an effect. So that's kind of the use of the t-test. So here we have an example. You want to know whether the mean petal length of iris flowers differs according to their species. You can find two different species of irises growing in a garden and measure 25 petals of each species. You can test the difference between the two groups using a t-test. So that means that you have two different types of species, and you can make, so you have two groups, 25 from one and 25 from the other. And then you set as I said, for doing this analysis, you need a hypothesis. So we have null hypothesis, and that hypothesis is that the difference between these groups is 0. So the null hypothesis is that group 1 and group 2, no difference, right? So my research question is there a difference between the mean of petal length according to their species? If this hypothesis is correct, then the answer is no, there is no difference. Okay, that will be the answer to my research question. And the alternative hypothesis is that there is, right? Yes, there is difference. So that's the hypothesis that you do when you want to test something. Always the null hypothesis is the one where there is no difference, where the groups are the same, or there is no effect, there is no relationship. And the alternative hypothesis is the one that tells you that there is difference. So let's move on to the next. So the t-test is a parametric test. So we need to be sure that the samples are independent, like the groups are independent, that they are normally distributed, and that there is homogeneity and variance. So make sure that the lengths, you can graph it to check if they are normally distributed, that the variance is not that crazy, either 0 or super large, and that both both samples are independent. And then the t-test is actually not that difficult, but a lot of mathematical formulas. But the t-test goes like this. First, you use the software will do it, but there is a formula to calculate a t-value. So with all the numbers that you have in your data set, the software will do the calculations and will end up with a t-value. That's the outcome of the formula. Then the software will check that t-value with a t-table that has some values, and the t-table tells you, like, if your value is around this, depending on your degrees of freedom. Degrees of freedom is only the number of observations minus 1. So in this case, it will be 50 minus 1, 49 degrees of freedom. So the table will tell you if your t-value is between this and this, that's good. You can reject the null hypothesis. So the software will check that, will check in the t-table where you are. And then another formula will convert that number to what we'll call the p-value. And this is the outcome that we care about. We are going to learn this in the quantitative lab, but the p-value will give you a number between 1 and 0. If that number is less than 0.05, we can say that we are going to reject the null hypothesis and we are going to accept the alternative hypothesis. That means that the two groups are actually different. Like there is an effect that depending on their species, the mean is going to be different, right? I hope it's clear. So all of this is done by the computer, right? The computer, right? And you should care about this, the p-value. If the p-value is less than 0.05, then we are happy, but we usually are happy because then the two groups are different. So we answer to a research question, yes, there is a difference according to their species. So that is the t-test. I hope that it's not too complicated. It's actually pretty simple. And that's everything. It doesn't tell you how strong it is. It only tells you that it is that the two samples are different, that there is actually a difference, and that's it. So you can do the same, but that I said that's why it's important. T-tests are good for like the thing with the diet, like one group before diet and the same group after diet, and then you do a t-test before and after. And if the p-value is less than 0.05, then we say that the diet had an effect. Of course, it has some limitations because in the case of and this works really well in biology and this kind of sciences like natural sciences, because then you can exclude a lot of things in a lab and you can only test one thing after another, because in social sciences it's difficult, even in the diet. If people did the diet, there is probably something more in there that only the diet, right? So maybe if you conclude with only the t-test that the diet had an effect, it has its limitations because probably the people in that group also did other things, like maybe exercise or maybe, I don't know, maybe they drink more water or something like that. So then those are kind of the limitations of the t-test, but it's a good start. And as I said, I have friends in physics, I have friends in biology that they use only t-tests because they are able to isolate everything and only test two things. But in social sciences, it's a bit difficult, but it's a good start. Then here we have another example. I like the example with the diet. Then we have the MindQuality test, which is kind of the same thing, but when you don't have these assumptions, when something is not in there. So in this example, we want to test if there is significant difference on attitudes toward gender discrimination and gender. So my research question is there a significant difference about attitudes towards gender discrimination depending if you are male or female? That's my question. So you can do a t-test, but it doesn't work because gender doesn't have normal distribution, right? Gender is 1 or 2, so the distribution will look something like crazy, right? Like you have the frequencies in here, and here you have 1 and 2, and it will look something like this, right? So the graph is this. So it's not normal distribution. So because of that, instead of using the t-test, you will use this. What is inside of the computer? I didn't write it in here, but it's basically kind of the same thing. So in the end, what matters is the p-value. I think I'm being very irresponsible by not saying what happens in there, but this is not a statistics course. So in the end, it's important that you understand how to interpret the MindQuality test and when to use it. So this is the great example when you have a a variable that is not scale, like gender, and then you want to test if there is difference between a scale variable, which is attitudes towards gender, and gender, not scale variable. So that is, I think in another video I'll explain the chi-square test.